All right, welcome to podcast 17. In podcast 16, we learned about the actual physical structure of the cell membrane, how it's built, phospholipids, proteins embedded, and so on. In this podcast, we are going to learn about what one of the most important things the cell membrane does, and that is to allow things to move across it. And that's the focus of this entire... There are many functions of cell membranes. Please understand, this is not the only function of them, but this is probably the most discussed and one of the most important functions. They control what moves from one side to the other. The way they do this can vary, but the fundamental thing to keep in mind is that molecules are always in motion. So a cell is in a sea of molecules that are moving all around it. Some of them are inside of the cell itself, some of them are outside of the cell, but they're moving. And what cells seek to do sometimes is just take advantage of that movement and let happen what would naturally happen, and sometimes cells try to control that movement and force that movement to go in particular directions. Okay? So that said, there are two basic types of transport across a cell membrane. One of them is passive, and that means the cell basically has to do nothing. This kind of movement is just going to happen whether the cell is doing anything or not. So no energy is required. Basically, all the cell has to provide is little openings, little windows, in order to allow this kind of movement to occur. Okay? In this type of movement, the molecules that are moving from one side of a cell membrane to another are moving with their concentration gradient. And what this means is, if you'll recall from first year biology, substances tend to move from areas where they are in high concentration to where they're in low concentration if they're just moving randomly. Okay? And in passive transport, where the cell is really doing nothing, it's just providing an opening for allowing molecules to just follow that natural flow of kinetic energy from high concentration to low concentration. Active transport, however, is quite different in that the cell is working against a concentration gradient. There is some concentration gradient that is not flowing in the direction the cell wants it to go, and so the cell is going to have to physically use its own energy to make those molecules move in the opposite direction of where they would naturally flow. It's like trying to push a river back upstream. It's going to take a lot more effort than just allowing it to go where it wants to go. Okay? Now, diffusion is what I hope might have come to mind right away when I mentioned this idea of movement from high to low concentration. If I open a bottle of perfume in one corner of the room, eventually people in the opposing corner would likely be able to smell it, because those perfume molecules move randomly, and they're just going to randomly move until they are able to be sensed all the way over in the other corner of the room. Random molecular motion. As you can see in the diagram, the high concentration of molecules is on this side of the membrane. Their random motion would eventually take some of them from high to low over to this side until the point where they are in equilibrium. Now, when these molecules reach equilibrium, that doesn't mean the molecular motion stops. Of course, these molecules will continue to move. It's just that the cell, nor you and I, would be able to discern a difference, because the molecules are still randomly moving, but there's no greater amount on one side than the other, so we're not really going to be able to perceive a difference from one side to the other anymore. Okay? So that's equilibrium. And diffusion, high to low concentration. Okay? This is a form of passive transport. The cell has to do nothing. This kind of movement is going to happen whether the cell is doing anything or not. The cell just needs to allow an opening so that these molecules can pass through. Okay? Now, osmosis is essentially a particular type of diffusion, specialized diffusion that involves water. Okay? It is the movement of water only from an area of high to low concentration. Water is such a ubiquitous, meaning it's everywhere, compound. Cells are always in water, so discussion of water kind of has its own little category of vocabulary and importance. So osmosis, the movement of water from high to low. As you can see in this diagram, well, let me not get into that just yet. Let me focus on this first. Also, generally speaking, osmosis is talking about the movement of water across a membrane. So in this case, the water molecules would be moving across this phospholipid bilayer. Usually, osmosis is not just movement of water molecules for the sake of their movement. Usually they are moving across a cell membrane, meaning this is going to be generally discussed only in terms of cells. Okay? All right. Now, there is another type of diffusion called facilitated diffusion, and this is the type of diffusion that is most often going to occur in cells. And what that means is most molecules, unless they are lipids or some type of little non-polar molecule, cannot just pass squeaking in between the little phospholipids. Most cannot do that. Most molecules need some kind of gateway, some kind of window, some kind of door that's going to allow them to pass through. Okay, so facilitated diffusion is still passive. It is still relying just on diffusion, the random movement of molecules from an area of high to low concentration. But they can only do so through these little selective doors in cells that are made by proteins. Okay, these are protein channels. So facilitated just means help. We're going to help the diffusion occur because we're going to get a little hole through which to happen. No energy is required from the cell nonetheless. Okay, now high to low, just like any type of diffusion. Okay, with the concentration gradient. These are terms that concepts you need to be very comfortable with. With the concentration gradient means going with the flow from high to low. Okay. Okay. Now, active transport occurs, as we said, when cells need to move some molecule against its concentration gradient, against the flow, in this case from low to high. Okay? Proteins are going to be involved in this as well, but they're not just going to be little tunnels, because the cell is going to actually have to force molecules to go in a direction they would not naturally move. Protein pumps. These are proteins that, generally speaking, use energy to undergo some sort of shape change, or what is called a conformational change. Okay? Shape change is also called a conformational change. Okay, and so when energy is applied, let's say to this molecule here, this is the protein pump. When energy is applied to it, it opens up and accepts a molecule, and then it might close up and force that molecule to the other side. And so this opening and closing would be caused by energy being applied to this molecule, and it then could force the substrate of concern towards one particular direction. Now I hope you'll notice this diagram is not a very good one because it's still showing this in high concentration to low, but in fact this would be, we would need to have this be the high side over here, or else we wouldn't need active transport, so that we would be moving these molecules from a low to a high concentration against their concentration gradient. Okay? Alright, low to high. Now, this does cost energy. Something we need to make sure we have under our belts is this idea that ATP is the energy currency, the energy money of the cell. ATP is what the cell spends when it needs to do something. We will talk much more about ATP a little bit later on, but for right now, understand it's a molecule that provides energy to the cells, and it has to use it if it wants to get anything done. So these little proteins, in order to change their shape and move these molecules, would be using up ATP quite a lot. They'd be using quite a bit of it. Okay? All right. Conformational change. Okay. One very well-known type of protein pump is called a sodium-potassium pump, and I wouldn't take too much time to understand all the nooks and crannies of it. We'll talk about this more when we get to nervous system stuff, but it is a classic example of a pump. It is extremely important in as nerve impulse transmission, which is why we'll talk about it when we get to the nervous system. But generally speaking, the sodium-potassium pump, if this is inside of a cell and this is outside, it is going to force certain charged particles into a cell and keep certain charged particles out here, so that we create what is called a voltage gradient, or a change in a difference in how much charge is on the outside versus how much charge is on the inside. Maybe the outside is more positive and the inside is more negative, and that polarity is going to allow nerve transmissions to occur. You don't really need to know all that, so why did I say it? Because I don't know when to shut up. But we'll look much more at that when we get to the nervous system. Just to understand, again, it would use ATP, it would change its shape, it would force sodium ions to one side and potassium ions to another side of the cell, and it would do all of this against their concentration gradient. So very energy intensive. All right, now cotransport. Essentially in cotransport, there is some substance we really want to move. There's another substance that we don't care nearly as much about, but we're going to use that substance to move the other. Okay? So I want you to understand this is going to be a combination of active and passive transport in terms of this facilitated diffusion idea. Okay? So we'll have a pump coupled with a transport channel. Okay, there's a pump that's going to use energy coupled with a transport channel that doesn't really use energy. Okay, so here's how it works. Okay, this is an example of how plant cells can get sucrose, sugar, into cells that already have lots of sucrose, sugar. Okay, imagine a plant wants to store sugar in its roots. Its roots function is to store sugar, hence there would be lots and lots of sugar already in those roots. But we want to get even more in because we're going to store it. Okay, so this sugar would not naturally flow in, okay, because it's in a lower concentration out here than it is inside the root. It's in a very high concentration inside that root where it's being stored up for use much later on. So what we're going to do then is not think about sugar right now. We're going to think about something else. I'm going to go up here. There are protons inside cells. There's piles of them. There always are. We don't need to know why. There just are piles of protons inside cells. And additionally, in membranes, there's often pumps called proton pumps. And these pumps will pump protons in one direction or another. In this case, this proton pump is pumping protons out of this cell into the outside environment of the plant, and not outside, but like out of the cell into surrounding tissue. Okay, so we're getting lots and lots of hydrogen ions out here because we're pumping them actively out of the inside of the cell. So we're building up a high concentration of hydrogen ions out here. Okay, now if diffusion is true and things seek to move from an area of high to an area of low concentration, then these being in high concentration out here would love to be able to flow back to where they would be in lower concentration. They want to flow from high to low. Well, they can't because we haven't given them a door. We are not going to let them pass through this bilayer. They're ions. They're going to love those little polar heads, and they're not going to go anywhere. They're going to get stuck out here. Okay, so they can't flow through the membrane, but they can if we give them a door. Down here is a door. Okay, but this door has a trick. It says, I'll let you in, hydrogen, so you can go and complete your high to low concentration gradient that you want to do, but I'm not letting you in unless you hold hands with sucrose over here, okay, and bring them in too. So what happens is this hydrogen ion, these hydrogen ions want to, the pull for them to flow back in is very strong. It's a steep concentration gradient. It's just like starting a ball at the top of the hill. It wants to roll down. Well, these want to flow with their concentration gradient. But this door will only work if there is a sucrose molecule nearby as well, and they will both fit into these active sites and both be will flow in at the same time. This will drag essentially this guy with him. This concentration gradient is so strong that when he couples with this guy, he will essentially drag him in against his concentration gradient. Remember, sucrose is already high in here, he's low out here, but this concentration gradient is so steep, he's just going to pull this guy in with him. So that's co-transport. We are going to couple this active pumping with diffusion, passive flowing of the hydrogen ions back in. We're going to link that up with pulling sugar in against its concentration gradient. That's co-transport. Okay, and I'm sure there's lots of words here. It creates, let me just let you kind of get a sense of this. Creates a high concentration of hydrogen ions, active transport, that's talking about this part, and this is simply saying that if it could, the hydrogen ions would want to flow back through, but we're not going to let them get through that cell membrane. Okay, so in plants, this is talking about the sucrose down here. It's in very lower concentration here. It's already in a very high concentration inside the root, so we're going to couple the flow of this with that, and this is going to essentially drag the sucrose through that special little transport channel, okay, against its own concentration gradient. Okay, so it's a lot of words. It's just repeating what we've already said. Okay, if you need to see this PowerPoint, you can go to our the class webpage and look under the PowerPoint link, and the PowerPoint is there in its entirety. All right. Passive transport has essentially, I'm just going to put all of this up here so we can kind of see what we're talking about. All right. We've got passive transport, simple diffusion, which might also include osmosis, okay? Facilitated diffusion, where we, actually that's a lie. Simple diffusion of non-polar hydrophobic molecules, meaning like lipids or oxygen, which is non-polar. We discussed that already, and it's very tiny. It can basically, these are things that can squeak right in between phospholipids. Eek! can get right in between because they're either tiny or they're nonpolar, or most likely both. Okay? Facilitated diffusion is polar hydrophilic things that couldn't get through this membrane, but they can if we give them a special door through which to go. And then active transport, where we are going to use some kind of pump and a whole lot of energy to move things against the flow of their concentration gradient. Okay? Nice summary here. I think you've got this on your handout. Simple diffusion, moving from high to low. These would be lipids and things like that that don't even need a door. They can just go on in between phospholipids. This is also diffusion, but it's facilitated because we're going to provide a door this time because these molecules can't squeeze in. Okay, and this is active transport, moving from low to high concentration using energy. Okay, and also a protein pump of some type. All right, now there are some molecules that are just really big, big hunks of food perhaps that cells want to take in, and these aren't gonna, you can't just allow these to diffuse through a single protein. So what we do is we employ some new form of an active type of transport called endocytosis, where we're going to essentially create a big bubble and just take in a large amount of food. You need to think amoeba for this. Okay, here is my amoeba right here. Here is a food item. It's right there. It's going to come up and take in the food item and enclose it in this little space. Okay, so these would enclose around that food particle here, and you would end up with the food contained in a little thing called a food vacuole. Endocytosis can take two forms. Phagocytosis, in which basically we are talking about solids being taken in, large solids. Penocytosis is taking in large amount of liquids. Okay? Exocytosis is getting rid of large amounts of substances. Usually we're thinking waste, but sometimes it is secretion of something useful outside of a cell. But in this case, let's presume these red molecules are waste products. They would be packaged inside a vesicle, and the vesicle would make contact with the outer membrane, the cell membrane, and then it would simply release the products or the waste to the outside. So it's a great little diagram showing phagocytosis. This is the taking in of a solid food substance, and you can see this food vacuole is made. So that's a food vacuole. It would fuse eventually with another organelle called a lysosome, and the lysosome contains digestive enzymes. Okay, and these would fuse together, and these enzymes would cause this food to be digested, and then it could be absorbed by the organism. Penocytosis is just taking in not a solid mass but solutes dissolved in liquid. Now both of those are generally not terribly specific processes. What's out here, if it's within the range of these pseudopods, this engulfing structure, is going to get taken in. So it's not very specific. This is quite specific and generally takes in only certain molecules, and it is called receptor-mediated endocytosis. These little structures here are called receptors. Okay, and usually these are proteins, perhaps paired up with some carbohydrate structures that act to receive certain molecules. And you can see these receptors have a very specific shape. These guys will not fit into the receptor, but these guys will. Okay, and so these receptors will collect these molecules, and as they do so, this little region of the cell membrane would begin to pinch in. This region that has the receptors is called a coated pit, and as the coated pit gets full of material, it will pinch in and package the material in a vesicle. Now, did it exclusively get just the specific material made for those receptors? No, it did pick up a few other things too, but generally speaking, it's going to grab more of what it is supposed to grab and take that into the cell. These materials that actually fit the receptors, those are referred to as ligands. So the purple triangles here, generally speaking, are ligands. One example of a substance that is taken into cells in this way is cholesterol. Okay? So in this case, cholesterol is a specific example of a type of ligand, something that would fit in with a particular receptor on a coated pit and be taken into a cell in this fashion. Okay? So this is very specific. There's that word ligand. Okay. All right. Cholesterol. Got it all. All right, and that's it. We're going to talk about water in some detail next, but that's it. Have mercy. Thank you. I appreciate your patience, and I will see you next time.